So I'm very honored to be here to give this talk, and I hope you enjoy it. So I'm more like telling stories and with the science as a background, but and how Steve impacted my career. So let's see how Steve Arnold helped me. So yeah, I'm gonna tell a story, and I'm not as good as Georgie Cham at PhD Comics, but I'm gonna use some of his stuff to tell a story. And oh, it's cutting off. Yeah, I need to fix this. Not sure how.
Is it too small?
Yeah, it's too big. It's cutting off the
I know
lower.
what it's done.
Oh yeah.
I think it wants to.
Yeah, maybe the slideshow. Yeah, great.
There.
Thank you. So, and of course, whoa, now it's moving along. Okay, it's moving by itself. So, of course, the hero of the science here is Steve. And I'm glad it is him. But there's some other characters, of course. So the first character is from the University of São Paulo. It's called Carlos. He's here, and I'm back here. And this is the beginning of my master's dissertation research. And all these people were part from the lab or previous people from the lab. And some of them collaborating now. And on this lab, we were interested in ecophysiology and evolutionary physiology. So Carlos had worked at the Andes with frogs and some physiological traits of the frogs. But he also had a line in the Brazilian Caatinga, which is a semi-arid environment that I really like. And, you know, there's frogs and toads over there, but rain is very unpredictable and temperatures are very high. So it's kind of an interesting environment to study. So I decided to work on that. But then, of course, everyone on the lab told me to read this paper, which is the Arnold paradigm. So this is my first encounter with Steve, actually. And I love this paper, and I really like this idea that performance is mediating the effects of morphology or behavior or physiology on fitness. So I was kind of impressed with this framework. And at that time, I really didn't understand all of it. So just this main idea that, you know, performance is important and it's part of the evolutionary process. So this was my first encounter with Steve. And then I decided to study thermal tolerance on tadpoles from the Brazilian Caatingas. And so I went and did all the field work. It was very cool. I really liked that part. But I also had some meetings with Steve's friends, right, still on paper. So, for instance, I'm very sad Ray is not here. But he was also quite an influence with this idea of the optimum that you can have and a thermal performance curve. And also Joe's work, you know, independent contrast. So it was really something that evolutionary physiology was paying attention to, that you need to take into account that species are not independent. So these papers was also a huge influence, and they're all Steve's friends. And then there's more Steve friends. Then I started to be interested in phenotypic plasticity and working with the tadpoles. So I tried to read some papers. And at that point also, you know, I was scared of the equations, but still I was trying. So I was trying to read a lot of evolution papers and not really getting the math, so I was feeling a little uncomfortable, but I tried to get better at that. So maybe not understanding all that much of the mathematics, I could do my work. So I actually compared species from Caatinga and Atlantic forests, and I did some thermal tolerance reaction norms to kind of measure the plasticity. So something came out of my master's, even though I didn't get all the theoretical and mathematical stuff underlying everything. So that was like a critical realization for me. You know, I was good at simple math, I think, but I was pretty sure, you know, at advanced math, I needed to get better. So I decided to change labs and try to really understand better mathematics and linear algebra and all this stuff that you need to know to study evolutionary biology. So, quantitative genetics. So the second character is my PhD advisor, also from University of São Paulo, Gabriel Marroig. And I did some courses of his, and I got really interested in this concept of modularity, so it was like something that really called my attention. So, and this guy here, Artur Porto, he was from my class, and at college. So it was nice to see his work, and I got really interested with the idea of modularity. And it was a mammalian evolution lab, but I went there to work with toads. And Gabriel accepted it, so I was really happy with that. So the idea there is more related to phenotypic integration and modularity, but of course that's related to the G matrix. So as Adam's already showed this picture, the shape of the G matrix really gives you a hint about phenotypic integration and how genotypes map to the phenotypes. So I really started to read even more evolution papers now on quantitative genetics. And there were some people from the lab that really helped out. So, people that knew more mathematics than me and knew how to program. So it was a very interesting period for me to learn all this stuff. Especially this guy, Diogo Melo, helped me a lot with programming and mathematics, and this guy. But, we had a lot of interesting discussions in the lab. So there were 8 of us doing PhD at that time, and we were 2 master students. So it was like a good environment to just, learn a lot. So it was great. So then I had, of course, more encounters with Steve and friends, all these papers. I like all of the papers, I really do. I like the idea of constraints. I like the G matrix. I love the adaptive landscape. I know Steve does too. And more, we discussed all these papers in the lab, man, and it was very good to have people that understood the mathematics so they could help me out to getting better at this. It was not just Steve and friends. Of course, I read other stuff, but it's all people that Steve knows, right? So I read a lot of Chevron stuff and Felsenstein stuff, Hansen and Hull also. So all these things related to the G matrix and also divergence, phenotypic integration. So I went on to do my PhD, right? All the parts of the work to finally get a thesis. And also, I was a mom at that point, so, 2 different projects. It's a lot of good stuff, and sometimes, I got a little tired. It's okay. So, really enjoying doing my PhD. And what I did, so I was working with the toads, and I had to have access to the skulls because I was interested in skull evolution. So I did a lot of CT scanning, like a lot. So this is showing, the toads and CT scanner to get access to the skulls. And I was working with this complex of species, Rhinella granulosa. Really interesting to study, and some of the species are from the Brazilian Caatinga, which an environment that I really like. And so I went on to estimate phenotypic matrices, so with the skull traits. So this is just showing the linear distances that I use within bones, and I was trying to understand what shapes trait covariation. So trying to connect, of course, the theory of morphological integration with quantitative genetics, right? So, just to give a quick idea, so integration, you're like focusing on relations of traits within modules. So they're more highly correlated within modules than between modules, and modularity is more focusing on the semi-independence that you can have in evolution when you have modules. So I went on to test that using neurons, which is really interesting because they have metamorphosis, right? So they have a cycle that is biphasic, an aquatic phase, a terrestrial phase, and I had this hypothesis that since I'm studying the adult skulls, that functional modularity related to adult functions of the skull would overcome the developmental signal modularity that is related to the origin of bones, because metamorphosis kind of could mess up all that signal, right? Because you have a lot of remodeling of the skull. So I tested this, created some competing modularity models for trait correlations, and I had this idea that the functional model would be the most likely one. And it is actually, but it's not really related to metamorphosis, so I can still detect developmental modules, but size variation is something that obscures that. So it was not because of metamorphosis, but still functional models were the most likely one. So that was a cool part of my PhD that I done. But also, since I estimated matrices for all these species, and I saw that they were really similar, so it got me thinking about, P matrix stability, and if the P matrices resemble G matrices, so G matrix stability, and this idea that why are they stable? So maybe there's a stability of the selection processes that shape those matrices. So I was really thinking a lot about that. So this is just representing some matrices of the toad species that I estimated. And of course, in quantitative genetics, you do have hypotheses for how are trait correlations created, right? So there's this Rydell hypothesis that it's a multivariate stabilizing selection favoring traits to go together if they are performing the same function. So really, again, calling me attention to function. So, I got really interested in it. I was thinking a lot about this. And then I went to Evolution meeting, so my first international meeting, and I met Steve. So this was kind of funny because I was waiting for Steve to stop talking to some guy like for half an hour, and I was like, ah. And I was not giving up. I was like, I'm going to talk to Steve, man. So I waited. And of course, he was tired, right? He was talking for half an hour with that guy. So I still went on. I said, hi, you're Steve Arnold, right? So I read a lot of your papers. I really want to talk to you. Are you tired? He's like, a little, but we can talk. So we talked a little. And then he said, don't you want to have lunch tomorrow and we can talk more? And I'm like, yeah. Can I call some friends? Because everybody wants to meet you. He's like, yeah. So I showed up with this crew for lunch with Steve. But they were so happy, man. And Steve was happy too, I hope. He looked happy. And then, we discussed a lot about the adaptive landscape. So it was really interesting. So these guys are from Chevrod's lab. This guy was in Scott Steppen's lab, and these are all people from my lab, from Gabriel's lab. So, it was really cool. And then, I learned that Steve is this very lovable guy, as Patrick was saying. So got me thinking, that maybe I could work with him, but I was not still thinking a lot about that. But yeah, so I came back, to the first paper that I knew from Steve, and then these modularity stuff that I was interested in, and I was thinking, maybe we could put some modules into Arnold paradigm, so it would be something like this, right? Instead of all the traits affecting different functions, some traits affecting more a main function and other traits affecting another function. And they will be two modules, but you still have the performance gradient here, mediated fitness, right? So then I got, an idea for working with Steve, maybe. So I started thinking about a postdoc, and then there was this evolution meeting in Brazil, which was pretty awesome. And everyone that I want to work was there, right? So I wanted to answer this question. So how does selection for functional performance contribute to trait covariation? So getting away a little from development and focusing more on function. And so I thought that lizards might be a good model for that because they're a very good model for performance. And so there was these two people here from Brazil that works with lizards. And Tiana was actually from Carlos' lab, from my first lab. And Steve was there, so, I joined everyone and discussed some ideas to try to study this. And Steve was kind of willing to accept me as his postdoc, which was great. And then, so I got funded for my postdoc, finished my PhD, and the first thing that I did was go to this Evolutionary Quantitative Genetics tutorial at Knoxville in Nimbius, I don't know how people like to say it. Very good course. So, and then I got some tools, especially with R, right, to work with everything that I wanted to do. So this is me over here, and Anna was also from my lab, and Joe was online. I was a little bit sad about that, but still, it was just kind of cool seeing his big head on the monitor. But it was just that, you know. It's like shown here on the picture. So it's pretty great when he got in. I was like, wow. And it was a great tutorial to do. And at that time, which was very interesting, I was working on a paper, you know, discussing with reviewers, and I was doing all the quantitative genetic stuff. So, you know, testing for selection or drift. And so I found that, you know, selection is the main process that shapes coevolution in the toads. And then, you know, doing eigenanalysis with the matrices to try to figure out which traits were under selection and if there was a relation with climate variables, so using comparative methods. So everything that was being discussed on the tutorial, it was exactly what I was doing at that point. So it was great, you know. And one of the reviewers, so I reconstructed selection gradients on the phylogeny, and I wanted to see if the evolutionary response were aligned to selection or maybe suffered some constraints, especially because of too much variation on the first axis of the matrices. But there was this reviewer that was like, yeah, you detect a selection and you're reconstructing ancestral traits using Brown emotion. Isn't that like a paradox? And I was really worried about that. So I remember that I asked in class, like, you know, what do you guys think about that? You know, and I remember that Steve Ginner said, you know, you can see Brown emotion as fluctuating selection also, and it's not necessarily drift. So I was like, yes, that's my answer for the reviewer. So it was great, man. The timing was great. So, you know, so the project that I wanted to do with Steve is actually try to answer this question using 3 species of lizards. So in the first part of my postdoc in Brazil, I CT scanned again, now lizards, to get access to the skulls. So it's pretty cool that one of the lizards is a really flattened lizard. So it's this one. And we had bite force, right? So we had a performance measure of this lizard, so I could get access to the bones, and now I could start to really try to make Steve help me why are bones going together or not, right? So the idea is that the bones that are participating in the same function will be more correlated, be suffering more correlational selection than bones that are not. So then finally I came here to Oregon State to work with Steve. You know, and I really like Corvallis. I really like this campus, so it was pretty great. Came as my son, so, you know, show him around a little bit. Really nice. Well, I had to see a lot of sports because I love sports, so we cheered for the Beavers. It was pretty cool this year. My son was really happy with all this, and me. And living in Corvallis is incredible because I'm from São Paulo, right? I'm not sure if everyone went there, but it's like a rock, you know, jungle, right? It's like just buildings, very gray, very loud, lots of people. So this is totally different for me and great. So, you know, I really liked living in Corvallis. I even did some kayaking in Steve's pond, which was also great. I had a backyard, you know, oh my God. I want that again in my life. I really do. And the thing is, we wanted to estimate stabilizing selection and correlational selection, so, you know, the gamma matrix. But in fact, it will be actually the functional gamma matrix. So I don't have fitness, unfortunately, for the lizards, which is very sad. But, you know, you can use performance as something that you can also estimate the gamma matrix. So that's what I came here to do with Steve. And so there's some Steve's wisdom that I started to get. So one thing that he said, you know, we were deciding which bones are we going to study, you know, sample size issues, how many bones can we work with to have some decent gamma matrices. And I remember then he said, you know, it would be really nice if you had like a functional model, biomechanical model of the bones. And then, so we could predict what the pattern would be and not just measuring and see if you find significant selection or not, but really where should I find it? So that was a very wise thing to say. And so I decided to work with the mandible bones, and we could have interpret it as a simple lever system and really try to predict where we would find correlational selection, really. So that's what we did. We had a very specific prediction of what should come out from the gamma matrices. And, using some eigenanalysis. So we had this prediction that correlational selection would favor bones that are part of the out lever or bones that were part of in levers, but not correlations between these two. So it's a very specific prediction, and that would be associated with stabilizing selection because I'm expecting an optimum for mechanical advantage in this case. So it was pretty nice to have this specific prediction. It makes your test stronger. So this became a paper that came out this year. And so we found some evidence for this contrast that I was expecting, especially in one of the species. So it is this contrast here, and it is under stabilizing selection. But for the other two species, we kind of found a different patterns, more directional selection on performance than nonlinear, and also a case of disruptive selection, which is kind of common in the literature, really, with performance. So got me and Steve thinking about, maybe disruptive selection is as common as stabilizing. I don't know, but it might be true. So it was pretty nice work to do with Steve. And now we're moving on to simulations, which is quite new for me. So I'm more of an empirical person. But, Steve has this with other people that he worked with, like Adam Jones, doing simulations. And so now we're working on this paper trying to answer how reliable is the estimated pattern of correlational selection. So, simulating a lot of sample sizes and different strengths of correlational selection and different distributions of fitness error and seeing how much can you trust the patterns on the eigenvectors of the gamma matrix, because so far significance of the eigenvalues has really been explored, but people are not really thinking of the pattern of selection, right, about the pattern that you get from the eigenvectors. So now we're working on that, and let's see how it goes. So more is Steve's wisdom. So while I was here, Steve thought it would be nice for us to teach a course, and it was a great idea, actually. And then he said, I don't think a lot of people in the department are interested just in modularity, so maybe it would be better we have a seminar that we discuss more things, because there are a lot of ecologists at OSU. So yeah, I accepted, and so we did a seminar on interaction networks and genomics, ecology, and evolution, and it was pretty great because there was these ecologists that participated in the course, so we got to discuss a lot about ecology and evolution, and that was not that common for me. So we had the participation of these guys, and we even had our private meetings trying to relate ecology to evolution, G matrices with ecological processes, and that's not so common out there. And then there's also Daniel, who was visiting from New Zealand, and so he got crazy about adaptive landscape. He's like, whoa. So it's funny, right? Because we as evolutionary biologists think everyone knows the adaptive landscape, come on. Yeah. It depends, So Daniel was like, wow, let's try to incorporate ecological factors in the equations of the adaptive landscape, and we're trying to work on that. So, it was really good to have this interaction here with more of the ecological people. So that's part of Steve's wisdom that I was granted with. And also, I participated in a scientific writing course of Steve's, which I really enjoyed. And he has, you had scientific writer partners, which is a great idea. So people that are generally do not know what you're doing are reading your drafts and saying this is not clear enough. So it was a great experience. And when I came back to Brazil, I decided to do a same format for our graduate course. So we did, and it was just postdocs that were running this. So one of the things that I like to do that Steve did in his course was talking about your paper, let's do a session here. So how are you feeling about your paper? People, were not always happy, of course. So it's a good thing to do, just people talk about their feelings on science and not just, what did you find significant or not, or, because it's really part of the science. So it was great. It worked, and students really liked it, so it was a good model to follow. So at the end, I want to say, I learned a lot with Steve, even before I met him, and a lot of other people too. You know, you start a conversation with a scientist when you're reading their papers, right? But then when you personally meet them, is even better. Of course, if someone like Steve, who is really open. And I think the thing that I most enjoy about Steve is just his enthusiasm for science. So for the animals, for the theory, for the mathematics. So I'm also very enthusiastic with this. So we got along pretty well. And, you know, after Steve, I have a lot of pictures of my son with snakes and newts, which I think it's also a pretty good thing to have. So here he was showing a snake to my son, and it kind of pooped. And my son was like, wow. And he's like, yeah, that happens, you know. And this is in Steve's ponds, and the kid catching newts. So it's really nice. So I'm very happy that I got to work with Steve, and we're still working. And I'm very happy to actually be here again, because I miss Corvallis. So thank you, Steve. I hope we continue our collaboration. And that's it. Thank you.